I'm starting again with an introduction to cells because I think that it bears kind of slowing it down a little bit so that you can really focus on these different key elements of what cells are and what they do. So right now you can see the two main types of cells that we've mentioned in class already, and that is a eukaryotic cell, eu meaning true, and prokaryotic cell, pro meaning first or early. And karyo, in both cases, is referring to a cell. And you can see that in these cartoon-like diagrams, you see a more complex cell when we look at eukaryotes. And when we look at a prokaryote or a prokaryotic cell, it looks much simpler, doesn't have as much stuff. Both of them have DNA, both of them have a cell membrane. But the difference, and this one here, you can see this prokaryotic cell actually has a tail-like structure called a flagellum. We'll talk about that a little bit. So these are some basic differences. Also, size is a big difference. So eukaryotic cells are typically much bigger in size than a prokaryote. Cell theory tells us that cells are the fundamental units of all living things. So if we went out, we can find any organism or anything that we say is alive on our planet is cellular. And we can look at it in a lot of different ways. One is that there are organisms that exist that are single-celled. That's all it is a single cell that then has the capability of growing and reproducing and responding to its environment. But a lot of the cells are not single-celled. They, in fact, build other things, such as in our bodies, cells of a certain type build tissue, and then those tissues build organs and organ systems. So here's the hierarchy. Cells are the basic unit, and for some organisms, that's it. And they don't go beyond that. But then others will have tissues. So some of the earlier organisms, some of the more primitive organisms have tissues but may not have organs. And then as organisms started to develop more, they develop. So from tissues of different types are used to build organs. And then organs of various types which work together form what is called an organ system. And then finally, all of the systems together form the entire organism. So this is called a hierarchy because it's going from the most simple form up to the more complex, or the most complex. The reason we've been able to understand cells at all is because we've developed microscopes that allow us to see them. And so this early type of microscope, which neither of these are. This is a light microscope, a compound light microscope. And we have some version of this in class. This is an electron microscope, which actually uses beams of electrons that are sent to a cell and give us an image. So really cool images. We've seen a few of them in our textbook. Here's an example, too, of something that microscopes and all things which are camera related, including your phone, have this ability to magnify, and they have an ability to resolve. So resolving power is looking at it as the ability to really focus in on detail. So it's not fuzzy. And then magnification is obviously getting bigger. You can see that here. We can go from this relatively small magnification, we keep getting bigger and bigger, and we get to see more detail. Here's resolution. And resolution, because it brings something into much better focus, it will distinguish two things which are right next door to each other. It can look at them as separate. So here you can see an organism called a radiolarian. And if we had a light microscope, this is what it would look like. If we use an electron microscope, we get all this added detail. So compound microscopes, they're useful. That's what we use. Some other types of microscopes, including ours, sometimes require that we use particular stains, some chemical stains, to allow certain structures to become visible. Here we go with electron microscopes. There are a couple types. Transmission electron microscopes beam their electrons right through the cell. But a scanning electron microscope, such as the one we're seeing up here, focuses on the surface of things. So here's some red blood cells. And in order to do that, they sometimes actually coat the object with something that will allow those scanning electron microscopes to send the image bounces back. Cell theory is this very important and underlying principle of all biology, and that is that all things are made of cells. Cells are the basic unit of life. And in order to get a cell, in order to have a cell come forth, you need a pre-existing cell. So only cells can make cells. All cells have 4 common components, no matter what type we're looking at. They're going to have a plasma membrane. Remember, that's going to be formed, typically it's a phospholipid bilayer. Remember our phospholipids. So in that bilayer part, remember that they have this, phospholipids are amphipathic. They have part of their one side loves water, one side doesn't. And so they organize themselves in this form of a membrane around a cell in order to either interact with water or not. Cytoplasm, which is whatever's in here, basically is cytoplasm, and cytosol is the watery portion of the cytoplasm. DNA, the genetic material, so here's our nucleic acid. And then here's our other nucleic acid, the ribosomes. Remember, those are the ribonucleic acids. Those are, an rRNA is a ribosome. We looked at mRNA, which copies information from DNA. And now we're looking at the ribosome, which is the type of nucleic acid that actually can synthesize a protein. In some cases, so in this picture, what we see is a single little circular loop of DNA here. And if this would be, and it would be representative of a typical prokaryotic cell, maybe a bacterial cell, what we would see is we'd see ribosomes, we'd see a cell membrane, they have a wall. This wall can contain something called peptidoglycan. We can also sometimes even see a capsule surrounding it. But we don't see any other membranes in here at all. As a matter of fact, this is not a membrane, that's just the DNA. So there are no membrane-bound organelles. So here is the typical features of prokaryotes. They lack membrane-enclosed or membrane-bound organelles. They have a cell wall that contains peptidoglycan. Prokaryotes are thought to be the very first cells, but they've done pretty darn well. So even though we could call them primitive, they are flourishing today. Many of them good guys. It's not all bad guys. And within that group are 2 big things called domains. So if we were going to take all prokaryotes and divide them into these things called domains, we'd see 2 of them. We'd see bacteria, which we've talked about more, and then another domain called archaea. So here again, prokaryotic, see the nucleoid is, instead of a nucleus, the nucleoid is just a region where this DNA is located. There's the dots are representing ribosomes. We can see in this, it's got a kind of a multi-layer exterior, because you see a cell membrane, it has a cell wall, this one has a capsule, sort of protects it. It can have that peptidoglycan, that material we said would be within the cell wall here. You can have little hair-like structures called cilia sometimes, but mainly what it has are these things called pili. And here's this big, long tail. Now, just to note that not all of these features are on all bacterial cells, such as this flagellum, for example. So you aren't always going to find them like this. And the other thing is that they aren't all shaped like this, and that doesn't talk about it here. But if it was this type of a shape, this is called a bacilli shape. So that's kind of rod-shaped bacteria. If it was round, it would be coccic or cocci. Coccus is the plural. So if you have like streptococcus, get strep throat, it means you have bacteria that have kind of this circular shape. This one, if you look at your yogurt on the back, you'll see that it contains lactobacillus, and bacillus is the name for this shape of bacteria. And then there's one called spirillum, and that's sort of spirally. But just so you know, they don't all look like this. This is very important, and understanding this is really important in biology, and that is that there's a reason that cells don't get huge, and prokaryotic cells in particular, but even in eukaryotic cells, there's a limit to how big a cell can be. So even an elephant or a great blue whale, may have bigger cells, but they aren't too big. And the reason why is because they need to move materials through the cell, and the cell isn't just a flat thing, it's three-dimensional. So we look at the surface area to volume ratio to understand this idea in this next slide. So here you see it. So here's a cell that has a surface or side rather of, like, we can say 1 centimeter or whatever unit we're talking about. And when we look at its surface area, which has 6 sides, we'd say that surface area is a 6. But its volume, remember, is length times width times height. So our surface area to volume ratio in this case is going to be reflected by 6. If it gets bigger, it increases in surface area and it also increases in volume, but relatively, the ratio between those two would be 3. And you can see here its surface area is 24, the volume is 8, so that ratio between them would be 3, which is smaller. And you see it and continuing. So as cells get bigger, that ratio decreases, and it's harder then for the cell, so everything comes in through the sides, right, through the surface, but it has to travel deep within the cell. And so as it gets bigger and bigger, it's harder to do. So keeping it smaller is helpful. Here you can see it again. It gives you a little bit bigger picture of that. And then we're going to stop.